Welcome back, everybody. Our next mini lecture in the reproductive series is going to be on the female reproductive system. Here's a very simple anatomical picture of a sagittal section of the female reproductive system so you guys can reacquaint yourself with the parts.
The uterus physiologically receives and nourishes the embryo. In anatomy, you learned it was comprised of a body. On top of the body was a curved portion known as the fundus, and below the body was an inferior portion known as the cervix, and the cervix projects down into the vaginal canal. In anatomy, you also probably learned that the uterine wall was 3 layers: an outer perimetrium, a nice thick middle layer of smooth muscle called the myometrium, and then an inner layer of epithelial tissue called the endometrium. We will come back to the endometrium in a couple slides because it is critical in the implantation of an embryo. The myometrium responds to rising oxytocin levels during childbirth to contract in response to that hormone. That is a classic positive feedback control mechanism. And contraction of the myometrium can also result from rising prostaglandin levels during menses or menstruation. And this contraction is known in layman's terms as cramps. The endometrium is classified as a mucous membrane because it is exposed to the external environment. So it's involved in protecting the body against the external environment. Like all mucous membranes, it is made up of 2 layers: a deep layer of connective tissue called the lamina propria, and a superficial layer of epithelium. The cells in this epithelium are long and columnar shaped, so this epithelial tissue is a columnar epithelial tissue. In addition to these 2 layers, there is a 3rd layer. It is called an endometrial or uterine gland layer. So it's a bunch of endometrial glands, which are also called uterine glands. These form as invaginations from the superficial layer of the endometrium, and they are responsible for producing the mucus, which will coat the epithelial layer in this mucous membrane.
The superficial epithelium is actually 2 layers. The deeper layer is known as the stratum basalis, or the basal layer. This is the portion of the epithelial layer that remains intact during menses, and it actually will give rise to the 2nd layer, which is the most superficial portion of the endometrium. It's called the stratum functionalis. The stratum functionalis is going to respond to increasing progesterone levels and will thicken up. It will begin to accumulate oxygenated blood and glycogen as we proceed through the uterine cycle. Now, the blood is going to come from arterioles known as spiral arterioles, and the glycogen is going to come from those uterine glands that I mentioned in the previous slide. It is the stratum functionalis that actually that degrades and is sloughed off during menses, or what is also known as menstruation. Now, the reason why the stratum functionalis is so important is that this is the layer in which the embryo will embed itself. This is the layer that is going to provide the growing embryo its oxygen levels and its source of glucose for ATP production, until the embryo can establish a connection, a more permanent connection, through things like the chorion and the placenta.
The process of making an oocyte is referred to as oogenesis, and it begins in females during fetal development. In early fetal development, primordial germ cells will migrate out from the yolk sac into the developing ovaries. And once they are there, they will differentiate to form diploid stem cells called oogonia. Oogonia is the plural word. Oogonium is the singular. Now, these primordial germ cells that migrate out from the yolk sac and into the developing ovaries are the exact same primordial germ cells that migrate out from the yolk sac and end up in the developing testes. So it is the same idea. In gonad development, you have primordial germ cells that migrate into the developing gonads. And depending on whether you are developing a testes or an ovary, they will form spermatogonia or oogonia. Either way, these cells are diploid stem cells. The oogonia are going to undergo several rounds of mitosis to produce millions of oogonia. And several oogonia are going to cluster together and become surrounded by flat epithelial cells called follicular cells. This is going to happen in the outer layers of the developing ovary. In other words, this is going to happen in the cortex of the developing ovary. Most of the oogonia within a developing follicle are going to degenerate through a process called atresia. So eventually what you end up with is a follicle containing a single oocyte. What you're going to end up with is a follicle containing a single oogonium. The surviving oogonia within an ovary are going to transform themselves to become primary oocytes within a structure called a primordial follicle. The primary oocytes are going to enter meiosis, however, they're not going to get very far. They're going to enter and stop at prophase 1 of meiosis. They're going to become surrounded by a layer of follicular cells to form what is called the primordial follicle. At birth, it is estimated that there are 200,000 to 1 million primary oocytes located within the ovary. These are found inside primordial follicles. At puberty, this number decreases to around 40,000. They are found to be inside follicles which are called primary follicles. And out of those 40,000 primary follicles containing primary oocytes, only 400 will develop over the rest of the reproductive life of the female. The rest of these will undergo atresia.
Upon the onset of puberty, the primordial follicle has transformed itself into a primary follicle surrounding a primary oocyte. Now, this mechanism of primordial to primary follicle transformation that happens prior to puberty starting is not very clear. We do know that primary follicles start off being made of a single layer of cube-shaped follicular cells called granulosa cells. These granulosa cells are going to divide to form several layers. And as they divide, they are going to produce a clear layer of sugars and proteins, so a clear glycoprotein layer that is going to get deposited between the oocyte and those granulosa cells. This clear layer of proteins and sugars is called the zona pellucida, and it's shown in these figures as that red line. So our primary follicle is going to be made up of multiple cells called granulosa cells, which are then going to be producing a zona pellucida, and then you're going to have the primary oocyte. The innermost granulosa cells found within the primary follicle are going to be firmly attached to that zona pellucida, and that cell layer is called the corona radiata. The reason why I mentioned the zona pellucida and the corona radiata is because they are going to play roles in fertilization. And our lecture on fertilization is going to be the last mini lecture in this series. So stick a pin in those two terms. Each month, several of these primary follicles are going to continue their development. And that development is going to be not under the control of hormones, but under the control of growth factors that are secreted by the primary oocyte. That subsequent stage of development is going to take that primary follicle to a secondary follicle. It takes, believe it or not, decades for a primary follicle to complete its development to secondary follicle and then to tertiary and Graafian follicle, because a primary follicle can remain dormant in the ovary for decades after the onset of puberty. So this primary follicle that you see here in figure C can remain dormant for decades until it is, quote unquote, selected to complete its development.
Outside of the granulosa cells that are found within the primary follicle, there will be the formation of two distinct layers of cells. These cells are called theca layers. They are the external theca and then the internal theca. When you see the formation of these two theca layers, you now have a secondary follicle, and contained within that secondary follicle will be the primary oocyte that is still halted at prophase 1. I think in this diagram, the secondary follicle is shown right there. So what you would have would be external theca, then the internal theca, then you would have multiple layers of granulosa cells, then you would have the zona pellucida, and then you have the primary oocyte. So that's the construction of our secondary follicle. Now this development from primary follicle to early secondary follicle, or what I should say is this development from primary follicle to secondary follicle, this can take almost a year, and it does not require any hormones because all of the growth factors required for transitioning from primary to follicle to secondary follicle are provided by the primary oocyte. The secondary follicle will now begin to accumulate follicular fluid within its center. And when that happens, you now have what is called an antral follicle, because the space into which this follicular fluid is accumulating is called the antrum. A lot of textbooks will refer to an antral follicle as a tertiary follicle because it's the stage after secondary follicle. This follicle is going to keep accumulating more and more fluid. It's going to become larger and larger and larger until it becomes this follicle structure right here, which is the most mature follicular stage called the Graafian follicle. This follicle will eventually contain the secondary oocyte. This is the follicle that will rupture upon ovulation. Now, the development from antral follicle to that mature Graafian follicle will take about 60 days, and it will require the hormone, follicle stimulating hormone, or FSH.
32 to 38 hours prior to ovulation, there will be a surge in LH, which is also called luteinizing hormone. This surge is going to be a result of a positive feedback loop that we're going to talk about in a bit. There's also going to be a surge of FSH right along with the LH, but we're going to concentrate on the LH because it is this LH surge that is going to result in ovulation. Several hours after luteinizing hormone peaks, the primary oocyte inside the Graafian follicle will leave prophase 1 where it has been stuck at, and it will complete meiosis 2 to split into two cells, a larger cell called a secondary oocyte and a significantly smaller cell, which I'm going to call a junk cell, which is known in textbooks as a polar body. Those cells are then going to rapidly enter into the next stage, which is meiosis 2. The secondary oocyte is going to attempt to complete meiosis 2, but it is only going to get as far as metaphase 2 and it's going to stop. The polar body is going to complete meiosis 2 and it's going to divide into two cells, both of which are called polar bodies. The secondary oocyte, like I said, is stopped at metaphase 2, and this secondary oocyte, which is halted at that meiotic phase, is the stage that is ovulated. The time from completion of meiosis 1 to ovulation of this secondary oocyte is anywhere from 18 to 36 hours. This slide concentrates on what is going on in the oocyte. So it's concentrating on oogenesis, but I will try and also relate it to follicular development. Remember, before birth, our ovary contains multiple primordial follicles. Each follicle is going to contain one primary oocyte, which is a diploid cell. So that is this cell right here. This primary oocyte attempts to complete meiosis 1. It only gets as far as prophase 1 and stops. Throughout childhood, our primordial follicles containing this primary oocyte are going to continue their development into primary follicles. So at puberty, you have primary follicles, each of which are containing a primary oocyte. Upon the onset of puberty, our primary follicle is going to begin its developmental stage into secondary follicle, the antral follicle, and then into the Graafian follicle, and the primary oocyte is going to attempt to complete meiosis. So let's concentrate on that. The primary oocytes located in the primary follicle are now going to begin their developmental journey. The primary oocytes are going to complete meiosis 1. They're going to split into two uneven-sized haploid cells, a secondary oocyte and a smaller cell containing leftover chromosomes and cytoplasm called the polar body. Note, we call it the first polar body. because both of these cells now are going to rapidly enter into meiosis II and attempt to complete it. The first polar body is going to get there. It's going to actually divide into two haploid cells, each of which contain one set of chromosomes, a little bit of cytoplasm, but they're junk. We don't want it. Our secondary oocyte is going to attempt to complete meiosis I. It's only going to get to metaphase, sorry, it's going to attempt to complete meiosis II, but it's only going to get to metaphase II. Now, while this is happening, our secondary follicle is growing into a more mature secondary follicle called an antral follicle, and it is maturing into the big fluid-filled Graafian follicle. So that when you look at a Graafian follicle, what you're going to see is that secondary oocyte, which has stopped at meiosis II, and two polar bodies. That is when ovulation happens. Ovulation is the rupture of the Graafian follicle. The secondary oocyte is going to be released, and the two polar bodies are going to be released as well. Now, I want you to note that the secondary oocyte is going to be surrounded by a clear glycoprotein layer called the zona pellucida, and a bunch of granulosa cells that have stuck to the zona pellucida. These granulosa cells are called the corona radiata. And if you remember, I mentioned those about 4 or 5 slides back. The bottom of the slide shows you what happens to the secondary oocyte upon fertilization. Fertilization is the union of egg and sperm nuclei to produce a diploid nucleus found within a cell called a zygote. In layman's terms, fertilization is the union of egg and sperm to produce the zygote. But fertilization requires the docking of a sperm onto the secondary oocyte. So here is the docking right there. It is this docking which will stimulate the secondary oocyte to complete meiosis II. So it will undergo the remaining stages of meiosis II. It will split into two cells of unequal size, a really big cell called the ovum, which has the sperm attached to it, and a smaller cell called a polar body. Now, if you count up all of the polar bodies that have been made, we have 1, 2, 3. Add to that the ovum, we now have 4 cells. That fulfills the rule of meiosis, which is 2 rounds of division, giving rise to 4 haploid cells. In the case of spermatogenesis, all 4 haploid cells develop into sperm. In the case of oogenesis, only one of those haploid cells is going to become the egg. The rest of these 3 cells, known as polar bodies, they're junk and they disappear.
There are lots of figures online that will show you the process of oogenesis, and we'll put it next to the process of follicle development. This is the best one that I've found so far online.
Fertilization happens within the first third of the oviduct, and it produces a diploid single cell called a zygote. As that zygote continues its journey through the rest of the oviduct and heads towards the uterus, it will begin the earliest stages of embryonic development. The earliest stages of embryonic development are going to be the development of the 2-cell embryo, the 4-cell embryo, the 8-cell embryo. And it's not a very fancy process, it's just mitosis. So our single zygote undergoes a round of mitosis to become a 2-celled embryo. That undergoes another round of mitosis to become a 4-celled embryo, an 8-celled embryo, etc., etc. 6 days post-fertilization, there is a very well-described and defined embryonic stage called a blastocyst. Here is a picture of what a blastocyst looks like at the bottom of the slide. It contains specifically a large accumulation of embryonic cells known as the inner cell mass. These are the cells that are going to continue embryonic development, give rise to the embryo, which will become the fetus, which eventually becomes the child. Okay, so our blastocyst is going to enter into the uterus and begin to implant in the stratum functionalis of the endometrium around 5 to 6 days post-fertilization. So let's say 6 days post-fertilization. Where it attaches is going to become the site of implantation. And that's going to be the endometrium, usually in either the fundus or down in the body of the uterus. But it is going to attach in a very unique way. It's going to attach to the endometrium such that its inner cell mass is oriented towards the stratum functionalis. And again, the reason why is the inner cell mass is the embryo. It is going to develop into the embryonic structures, and that embryo is going to become a fetus. At 7 days, the stratum functionalis at that implantation site is going to become highly vascularized. This is so that the developing embryo is going to be provided with a good source of oxygenated blood and also a good source of glycogen, which is going to become glucose, which is going to become ATP, which is energy you need to develop. Another 2 days go by, let's say day 9 post-fertilization, and the blastocyst will be completely embedded in that stratum functionalis. Now we're going to talk a little bit more about fertilization and embryonic development in the very last mini lecture. So stay tuned, we're going to talk a little more about it. The same 3 hormones that were responsible for control of the testes are the same 3 hormones that are responsible for control of the ovary. This is because these hormones were initially discovered and named after the female reproductive system. So the gonadotropins FSH and LH are named for ovarian follicles and the corpus luteum that develops after ovulation. And this is because the early physiologists that did work in this area were more interested in the female reproductive system than the male reproductive system, because females got pregnant and gave birth. So we're going to start off with gonadotropin releasing hormone coming from the hypothalamus, traveling down to the anterior pituitary and telling the anterior pituitary to release FSH and LH. FSH and LH will travel via the bloodstream down to the ovary. FSH is going to initiate the development of the last stages of follicular development. So it's going to initiate the development of the antral follicle developing into the Graafian follicle. Luteinizing hormone stimulates the rupture of the Graafian follicle in the process known as ovulation. And we are going to cover how luteinizing hormone does that in a minute. But I will tell you that both LH and FSH stimulate the secretion of estrogens from the follicle. So LH stimulates the thecal cells, which is the outermost portion of the follicle, to make testosterone. And then testosterone is taken up by the inner granulosa cells and converted into estrogen. And this conversion requires an enzyme called aromatase. So you may not realize it, but estrogen starts off as the hormone testosterone. We're also going to see that luteinizing hormone, again, triggers ovulation, and after ovulation, it is going to result in the development of the corpus luteum. In addition to gonadotropin releasing hormone, FSH and LH, there are 4 more hormones I want you to learn in association with the female reproductive system. That's estrogen, progesterone, relaxin, and inhibin. The growing follicle will produce large amounts of estrogen. These are called follicular estrogens. Again, refer back to the previous slide where I mentioned how FSH and LH are involved in stimulating the follicle to produce estrogen. Prior to ovulation, the follicle produces inhibin, and after ovulation, inhibin is going to continue to be released by the corpus luteum. Now, inhibin sets up negative feedback inhibition of FSH and LH. High levels of inhibin are going to prevent the release of gonadotropin releasing hormone, and then it is going to inhibit the release of FSH and LH. And no new oocyte production will result as long as these 3 hormones are shut down. And the reason why inhibin is made by the corpus luteum is, in essence, the female has to figure out whether or not she's pregnant. If you have confirmation that the female is indeed pregnant, the last thing you want to do is go through another round of reproduction and make more oocytes. So inhibin levels are going to stay high until pregnancy is confirmed or not confirmed. If not confirmed, the inhibin levels are going to drop and you will get a whole new round of gonadotropin releasing hormone, FSH and LH secretion. Lastly, relaxin. Relaxin is a hormone made by the corpus luteum post-ovulation. Relaxin does exactly as its name implies. It relaxes the uterus by inhibiting contractions of the myometrium. This is very important to the implantation of the embryo. The last thing you want to do is implant the embryo and then have the myometrium spontaneously abort it. High levels of relaxin will be maintained throughout pregnancy. The first levels are going to be made by that corpus luteum, but as pregnancy continues, it is then taken over by the placenta. The three types of estrogens are estradiol, otherwise known as beta estradiol, estrone, and estriol. Estriol is made at very high levels throughout the reproductive life of the female, so it is the most plentiful estrogen made, and it is the most powerful estrogen made. Estradiol levels will drop off as the female approaches menopause, and it will be quite low through menopause. And at that point, the most prevalent estrogen being made during menopause is estrone. It's still not made at very high levels because the female is menopausal. And estrone has a relatively weak effect on the female reproductive system. Estriol is only made during pregnancy, and it is the weakest of the three estrogens. Now, when an estrogen is made by a follicle, it's called a follicular estrogen. When an estrogen is made by a corpus luteum, it is called a luteal estrogen. The follicular estrogens are primarily estradiol. So, in other words, the major source of estradiol in the female is going to be the follicle, and then the alternate source of estradiol is going to be the corpus luteum. The function of estrogens is to promote the development of the female reproductive structures, the secondary sex characteristics, and the mammary glands. However, estrogens have other functions outside of the female reproductive system. For example, estrogens will increase protein anabolism, so they increase protein synthesis. And this is specifically seen in the skeletal system. These estrogens are excellent at promoting the synthesis of bone proteins. So it is known very well that estrogen maintains bone density and bone mass in females. Estrogens also lower LDL blood cholesterol levels. They also are thought to have cardioprotective effects. And this is why, as levels of estrogen, specifically beta estradiol, drop precipitously as women enter into menopause, things like protein synthesis, blood cholesterol levels, and the cardiovascular protective effects drop off quite significantly. Think of progesterone as the hormone of pregnancy. It is made by the corpus luteum. Its job is to increase endometrial lining thickness and to maintain the thickness of that endometrial lining so that the embryo can implant and survive within it during the earliest stages of embryonic development. Decreasing progesterone levels are going to be associated with the breakdown of the stratum functionalis of the endometrium and what is referred to as menses or menstruation. However, progesterone also decreases the maternal immune response. It is involved in uterine smooth muscle contraction. As a matter of fact, falls in progesterone levels is one of the triggers for the onset of labor. And finally, progesterone is thought to inhibit lactation. Progesterone plays a role also in breast development at puberty. It is thought to affect the migration of sperm through the female reproductive tract, although the mechanism of this is not known. Outside of the female reproductive system, progesterone is known to decrease fluid retention. It's thought to have a protective effect on neurons. By the way, estrogen is also thought to have a neuroprotective effect. And it's even thought that progesterone might play a role in addiction. Progesterone requires increased expression of estrogen levels in order for it to act. There has to be increased estrogen levels, increased progesterone receptors within cells. Decreases in estrogen and progesterone levels overall causes the thinning of epithelial tissues. This can lead to skin wrinkling and decreased vaginal lining thickness.
The female reproductive cycle is actually made up of two cycles, an ovarian cycle and a uterine cycle. The ovarian cycle will describe the events during and after the maturation of the oocyte, and the uterine cycle is a concurrent series of changes that happen in the endometrium of the uterus so that the uterus becomes prepared for embryo implantation.
This slide describes what happens at the follicular phase or pre-ovulatory phase in the ovarian cycle. It is the most variable of the ovarian phases. It can last anywhere from day 0 to about 13, 14 days. At the start of this cycle, there are very low levels of estrogen and progesterone. So at day 0 of the follicular phase, at day 0 of the female reproductive cycle, there are very low levels of estrogen and progesterone. And this is going to relieve the inhibition on the hypothalamus and the anterior pituitary. As a result, there's going to be a release of gonadotropin-releasing hormone from the hypothalamus and FSH and LH from the anterior pituitary. And we're not going to worry too much about what luteinizing hormone is going to do at first. In this follicular phase, we're going to concentrate on what FSH does. But I want to backtrack and remind you about something. At day 0, the ovary contains many follicles at different stages of development. There could be several follicles that are in their primordial phase. There are those that can be primary follicles, and there are going to be those that are secondary follicles. Again, reminder that this early development is the result of growth factors and not hormones like FSH. And this development starts at puberty, and it can take several months to years for a follicle to go from primordial to primary to secondary. So if you were going to take an instant snapshot of what the ovary looks like at the start of the ovarian cycle, you're going to find hundreds and hundreds of them are still primordial, hundreds and hundreds of them are primary, and then hundreds of them will be secondary follicles. As I mentioned in the previous slide, at day 0, we're relieving this inhibition on the hypothalamus and the anterior pituitary. We're going to get a pulse of gonadotropin-releasing hormone, and we're going to get a surge of FSH. And this surge of FSH, you can see over here, happens at about day 0. Okay, and it is going to have an effect on the follicles. FSH is named follicle stimulating hormone because of its effect on ovarian follicles. What it's going to do at day 0 is it's going to cause the continued development of several secondary follicles containing primary oocytes. So it's going to start that secondary follicle development. At about day 6, one of them is going to become the dominant follicle. The other ones are going to cease to develop, and they're actually going to degenerate. This dominant follicle is shown in this figure as the bigger follicles, this big follicle right here. So we'll call the one I'm circling the dominant follicle. It is going to begin to secrete estrogens, and you can see as a result, estrogen levels come up. These are follicular estrogens. And these follicular estrogens now are going to inhibit FSH and the development of other follicles. So it's like one follicle, one secondary follicle gets to be the dominant one, and it shuts down the development of all other ones that are competing with it. This dominant follicle is going to develop into a very large fluid-filled mature follicle that we're going to call the Graafian follicle. That Graafian follicle is going to continue to secrete estrogen. And the Graafian follicle, I am going to circle here in green. Here's our Graafian follicle or mature follicle. And again, you can see because of it, there is a dramatic increase again in follicular estrogens. So if you look down here, the estrogen being showed in the uterine cycle, kind of misleading because the early spike of estrogen you see in this chart is actually due to the follicle. They're called follicular estrogens. So I wanted to pull out again what's going on in the follicle from that previous diagram and show you about day 0 to day 6, you've got primordial follicles developing into primary follicles, or primary follicles just getting ready to begin their final phase of development. At around, between this day 0 to day 6 phase, you're going to have a nice surge of follicle stimulating hormone. And that's going to take our follicles now through the remaining stages of their development. The primary follicles are going to develop into those immature secondary follicles. The immature secondaries are going to go through a series of progressions where they're going to be a big fluid-filled secondary follicle. And then that secondary follicle is going to become the mature Graafian follicle that is just getting ready to ovulate the egg. A couple other things I want you to notice is that I pointed out the green layer that you start to see around the early secondary follicle stage. That is the glycoprotein layer known as the zona pellucida. And then you can see in the mature Graafian follicle, you have a really nice big thick green zona pellucida. And you can see that these cells right here are a little darker in color. Those are going to go with the ovulated oocyte, and that's going to become the corona radiata.
While the follicular phase is going on, in the uterine cycle, we have the phase known as menstruation or menses. This is the first 5 to 7 days of the reproductive cycle. It corresponds to the degeneration of the stratum functionalis of the endometrial lining. And it is actually started by events that take place in the previous reproductive cycle. The events that trigger the breakdown of the stratum functionalis are going to be the drop in estrogen and progesterone from the previous cycle. So this drop right here will lead to menses in the next cycle. The drop in estrogen actually results in the death of the cells of the stratum functionalis. So those die and basically degenerate, but the declining estrogen and progesterone levels don't affect the stratum basalis. What results is, over the course of the 7 days, a loss of anywhere from 50 to 150 milliliters of blood, tissue fluid, mucus, and epithelial cells from the stratum functionalis.
Ovulation is the rupture of the Graafian follicle and the expulsion of the secondary oocyte. And this usually happens around day 14. The ovulated secondary oocyte is going to remain surrounded by its corona radiata and zona pellucida. Ovulation is triggered by a positive feedback system. High levels of follicular estrogens are going to cause a pulse in the release of GnRH and the release of FSH and LH. And this pulse of GnRH and LH happens just prior to ovulation. Don't worry about the FSH, concentrate on the LH. The increased LH levels that happen because of this pulse induces the rupture of the Graafian follicle. This will happen about 9 hours after that peak levels of luteinizing hormone are reached. Now, this cannot happen without a specific level of estrogen. So we say that this is a permissive interaction between luteinizing hormone and estrogen. By the way, the rise in luteinizing hormone levels are the basis for at-home ovulatory tests, or what we call ovulation kits. There are some physical signs that can be experienced during ovulation. For example, the female will see an increase in basal body temperature. Changes in cervical mucus will happen. The cervix will soften. And some females will even report an ache or a sharp pain on one side of the abdomen. This is thought to be the actual event of ovulation, and in medicine, it is known as Mittelschmerz, which is German for middle pain. Following menses, the stratum functionalis layer of the endometrium is at its thinnest because it has degraded and has been shed. So in the proliferative phase of the uterine cycle, the stratum functionalis is rebuilt, and this is the job of follicular estrogens. Follicular estrogens are going to begin the repair of the stratum functionalis. It does so by stimulating the cells of the stratum basalis below the functionalis to undergo mitosis and produce a brand new stratum functionalis. So because these stratum basalis cells are undergoing mitosis to produce the new stratum functionalis, this is why we call this part of the uterine cycle the proliferative phase. The epithelial cells in the newly built stratum functionalis are going to lengthen to become columnar in shape. This is going to lead to an overall increase in thickness. However, the true increase in stratum functionalis thickness is going to come post-ovulation. That is going to be the job of progesterone.
After ovulation, we enter into what is called the post-ovulatory phase of the ovarian cycle, or what you could also call the luteal phase. This is the most consistent part of the reproductive cycle. It's the most consistent part of the ovarian cycle. It is 14 days. So the variability in the ovarian cycle comes at the beginning. It comes at the follicular phase. We've now undergone ovulation. The mature Graafian or tertiary follicle has ruptured, and it has released the secondary oocyte. In the luteal phase, the mature Graafian follicle collapses and begins to bleed. That bleeding follicle is going to clot, and it's going to be called the corpus hemorrhagicum. This clot is important because it is going to result in the transformation of this corpus hemorrhagicum into the corpus luteum. And this transformation is going to be under the control of luteinizing hormone. So that pulse of luteinizing hormone not only sets up ovulation, but begins the transformation of the corpus hemorrhagicum into the corpus luteum. The luteal cells of the corpus luteum are going to produce progesterone, estrogen, relaxin, and inhibin. So you can see, if you look down at the uterine cycle, you can see that the appearance of your corpus luteum starts to coincide with the peaking of progesterone and the peak of this blue estrogen line, which is going to be your luteal estrogens. High progesterone, estrogen, and inhibin are going to become the negative feedback signals for the inhibition of GnRH. So you can see I put in this ovarian cycle where the potential pulse of GnRH will occur to set up the next cycle. This is inhibited by increased levels of progesterone, increased levels of estrogen, and increased levels of inhibin. So that's your standard negative feedback.
Progesterone, produced by the corpus luteum, promotes the continued growth and the vascularization of the stratum functionalis layer of the endometrium. During the uterine cycle secretory phase, which is the phase post ovulation, the stratum functionalis can thicken anywhere to about 12 to 18 millimeters, and you can see it actually thickening exactly along the same pattern as progesterone levels rising. The reason why it's called the secretory phase is because endometrial glands located within the endometrium will begin to secrete glycogen, which can then be taken up by the cells of the developing embryo, broken down into glucose, and used for energy production.